And hello everybody. Hope you all enjoyed my new fancy countdown with horrible music. Yes, unfortunately rights-free music is not very good, but there you go. That's my attempt at trying to ease you into things a little bit, rather than just having me abruptly starting talking at about 20 seconds past 4 o'clock. So we'll see how that goes. But I mentioned on Twitter, and I thought I would show you here actually, that I made that countdown using a bash script. So because we've done our fair share of scripting and I thought it might be of interest, I thought I'd show you how it works. So for broadcasting on YouTube, I use a piece of encoding software called OBS. And OBS is capable of taking the contents of a file and displaying it on the screen. So what I've done is I've written a script which figures out the time between the current time and 4 p.m. Turns it into a minutes and seconds countdown, and then prints it into a file repeatedly. And I'll just quickly show you how that works. So let's turn that off. There you go. So here is my the script that I wrote yesterday. So I've written a function in Bash. We never covered functions, but essentially you can use this like another Bash command. It can take arguments. So in this case, it takes a single argument, which is a number, and it converts it into the number of minutes and the number of seconds left in a countdown, which is calculated in seconds. And then within a loop, and this is an until loop. So this loop runs until the number of seconds left until 4 o'clock is less than 1. So basically until there's no until 1 o'clock, until 4 o'clock. So the date today is calculated here, and then the number of seconds until 4 p.m. today is calculated here. And then within the loop, I calculate the current time and then do some maths to work out the difference between the current time and the time the time at 4 o'clock today. So this calculates the number of seconds left, and then this formats it into minutes and seconds and prints it into this countdown.txt file, which is then read by my encoding software OBS and printed onto the screen. So as the time proceeds towards 4 o'clock, it gradually counts down. I think that was quite a nice little exercise for myself to be able to do last night. So I thought I'd share that with you before we get going. A little bit of practical Bash. Okay, so yesterday was another one of those days where essentially we spent an hour writing one line of R, really. So we spent the hour yesterday getting our count data from the files produced by Salmon into our R session using the library, this TX import library, which we talked about briefly last week and maybe in a little bit more detail yesterday. So TX import takes the data that's in those files, interprets it and reads it and gets it into R, into a data structure in R, which we can then use within our R session. The other thing that TX import does is it takes those transcript level counts that are produced by Salmon that we looked at a little bit yesterday. We looked at those output files. We've looked at them before as well. So it takes those transcript level counts and it summarizes them at the gene level so that we can analyze differentially expressed genes rather than having to worry about differentially expressed transcripts, which is a more complicated question to ask. I might come back to why that's a more complicated question on another day, but I'm not going to go into it in too much detail today. So now that we've got that data in R, our next job is really to get that data from where it currently is in R and get it into the library which we care about for analysing that data, which is DESeq2. So I have talked about DESeq2 a little bit before, although not in a huge amount of detail. But DESeq2 is our platform for analysing this count data at the gene level, and fortunately, DESeq provides us with good methods for importing the data from TX import. The first thing we're going to have to do, though, is install DESeq2, because we haven't yet installed it. So we're going to go back to package installation again, which we haven't done for a little bit. Okay, so let's get R back. Sorry about that. There we go. Right, here we are. So yes, package installation. That's the first thing that we want to do. So if you recall from when we've done this in the past, the first package, pretty much the first package that we installed was a package called Bioinformatics Manager, and that's because that provides us with an easy way of installing packages, whether they're on CRAN or whether they're in Bioconductor. And that function is the install function of Bioinformatics Manager. So that's what we're going to use to install DESeq2 here. So we should have, if Bioinformatics Manager is attached to your current session, which for me it is because I've not restarted R since the last time that I installed a package, but if it's not, then run library Bioinformatics Manager to attach the package, and then we run install, and the package that we want to install is DESeq2, where the D, the E, and the S of DESeq are all capitals. And the capitalization does matter, as with almost all things in programming and at the command line, and as I'm sure you've discovered, and certainly I have discovered at various points, all of these things are the command line is, they're all case sensitive. And so if I try to install DESeq with the lower cases, then the install package is going to complain, he predicted. It's a strong prediction. Here we go. We're thinking about trying to install DESeq. And so it's gone straight to, there you go, try to update my out-of-date packages, and has said DESeq2 is not available, and that's because it's not capitalized correctly. And actually, Bioconductor is kind of nice, the Bioinformatics Manager package is kind of nice, and suggests to me what the proper capitalization is. But it's not going to try and install that for me unless I get that capitalization correct. So capital D, capital E, capital S. And this time it will go away and install it. And you can see there's quite a few dependencies to install as well. So this is going to take a few seconds to download all of these packages. The nice thing about doing this on Mac OS and on Windows is it downloads precompiled binary packages, which don't take that, they just have to be downloaded and put into the right place, and it works. If you're doing this on Linux, all of these packages would have to be compiled after they've been downloaded, and this would take an awful lot longer. So it's kind of nice that on Mac OS and on Windows, we don't need to worry about that compilation process because it's all been done for us by the good people at Bioconductor. So this is downloading all of the dependencies of DESeq and DESeq itself. It's DESeq2. There is DESeq1. I'm not sure whether the package is still distributed, but DESeq2 is as the name suggests, is the second version of DESeq. I will generally refer to it as DESeq because DESeq1 really isn't in use anymore. So where I say DESeq, I do always mean DESeq2. Okay, so that's the packages downloaded and installed. And again, I'm not upgrading those packages. It's asking me whether I want to upgrade because they're system-wide installed. And in order to get those to upgrade, I would need to run this as an admin user. Right, so we've installed DESeq, and what we now need to do is add it into our script. So make sure that we load the package within our script. So there you go. That's loading DESeq. You get a lot of chatter in the console when you load it as well. There we go. So that's loaded, which is good. And then to get our data from the thing that we got from TX-import, the results of our TX-import command into DESeq is actually really a straightforward process. It's just a single function because there are authors in common between TX-import and DESeq2. So it is in the interest of the authors of both of those packages to make them work nicely with each other. In order to go from the results of TX-import to a DESeq dataset, we just need to run function DESeq dataset from TX-import, which takes the results of TX-import, which is the count data, and then it takes a thing called col data. So count data here is our what's called our TXI. That's the object that's produced by TX-import. Then the other arguments of this DESeq dataset from TX-import are col data and design. And I'm going to talk through these two things because although this is we just got a single function here, it's nice to know what these arguments mean. So col data is sorry, I'm seeing a bit of chatter in the comments, which I'm just going to pause and talk a little bit to the people in the comments. So configuration failed for package XML. That's an interesting one, which I could solve on Linux really easily because it means libxml is missing on Linux, but I don't know how to solve it on, I presume, Windows. And Graham, you're missing an E in DESeq, so it's SEQ, not SQ. So yes, I can't really help very much more with those right now. I'm afraid what I'm gonna do is I'm gonna press on with what I'm doing and let Peter try and field those if he can. If you're on Linux, then install libxml. So at the command line, do sudo apt install libxml. It might be libxml2 or it might be libxml dev. but you need one of those to get the libxml headers to be able to install the XML package on Linux. So that explains that. Let me just find out exactly what the dependency is. Package XML Linux stats. libxml2 dev is the package. I'll just type that in the comments. There you go. That. Okay, so hopefully that will get people back on track. Right, so col data. So what is col data? Col data, as the name doesn't quite suggest, is the data about the columns in your count data. So the way that your count table is formatted is that I'm just looking for my whiteboard, which is not running. Doing well today. I think it's that one. No, not that one. Right, whiteboard, there we go. Right, so, okay. Sorry, I'm just, there we go. Stick that up there. Open up my whiteboard. Make sure it's running. And then we'll talk quickly about the way that our data is organized. Actually, I can show you the slide that I was going to show you in a minute. Right, so there we go. This is the results of TX import. Okay, so this is a little bit of a slice of a count table. So in this example here, we've got 6 samples and about 10 genes, I think, in this example. But this is what the results look like. And so the genes are in the rows and the samples are in the columns. And what DESeq wants when it asks for col data is the data about the columns in your data. So the columns in your data being the samples. So the col data is a description of the samples, which for us is our sample table. So let me just take that away again. We've got our sample table, which we created last week actually now. So if we have a quick look at that, better refresh our memory of the sample table. There you go, that's our sample table. This has the data about the columns, about the columns of our count data. So each one of these columns in this sample table is some information about our experiment, about each of our samples. I'm going to reformat this slightly to make it a little bit more friendly for DESeq. So there's 2 things that we need, a couple of things that we need to do, 2 or 3 things that we need to do. The first thing that I want to do is I want to remove any spaces from the column names, because that doesn't play nicely outside of the tidyverse. It doesn't play particularly nicely inside the tidyverse. So I'm going to remove the space from this sample name column header. The other thing that we need to do is turn it into a data frame from a tibble, because DESeq doesn't understand tibbles. So we'll fail if we try to give it a tibble. So we need to turn it into a data frame. And then the final thing that I want to do is I want to produce a column which has whether the sample is mock treated or SARS-CoV-2 treated in a column which isn't separated by spaces, and also is a factor. And the reason for that is that we want to be able to analyze these. This is a factorial experiment, so these whether something is infected or mock infected should be described by a factor, because it can either be one thing or another, and that's what factors are for. So we're going to slightly tinker with this data just to make it a bit more friendly for DESeq2. So the first thing I'm going to do is turn it into a data frame. Okay, so sample table equals as.data.frame sample table. And that turns it from a tibble, which it currently is, because you can see when I printed it out here in the console that it tells me it's a tibble. This will convert it from a tibble into a flat R data frame. So now when I print it out, it will no longer tell me that it's a tibble, and it prints out the whole thing for me. Okay, so that's one thing. The other thing that I want to do is I want to, there's two more things, sorry, the two things that I want to do. I want to change this sample name so it's not got space in. So in order to do that, we do col names, which is a function which allows you to retrieve the col names, but also set the col names. So the col names of sample table, and I just want the first one. Okay, so that's the first sample name, the first column name. I just want to change that. Instead of being sample name, I just want it to be sample. Okay, so this will change that column header, that column name. And now if we look at our sample table in our console, you will see that that column header has changed. So now it's just sample. And the other thing that I want to do is I want to add a new column, which is effectively like some of the things that we've already got in here. We've got treatment, which tells us whether it's mock or infected. And cell line, I want to introduce a single column with no spaces in, which is a single factor description of the whole experiment. What we're going to do is I'm just going to define this actually so we want our conditions. We've got 4 conditions. We've got mock NHBE. We've got infected NHBE. We've got mock A549. And we've got infected A549. So I'm assuming here that we're going to keep the cell lines separate from one another. That we're going to look at the cell line dependent effects of SARS-CoV-2 infection. You could keep this as 2 separate descriptions. So we could have a column which is mock or infected, column which is cell line, which we kind of already got, and then we could look at whether the infection has any cell line independent effects. But for now, I'm just going to keep these separate. So those are our conditions. But actually what we want to do is we want to repeat those, because we've described 4 conditions, but we've got 12 samples. So we want to repeat each of them 3 times to describe our 12 our 12 samples. So we use rep for that, the same as we did when we did these things last week. Conditions each equals 3 will then describe those in the right order. And then we want to, so we need to do a couple of other things. We need to turn that into a factor. So let's save that as conditions. So now that's length 12, which is the same number of columns that we've got, and let's turn it into a factor. There we go. Conditions, like that. So that's turned it into a factor. There you go. So it's got 4 levels, and we're going to add that into. We're now going to add that into our sample table as an additional column, because this is. So this is effectively, this one column is effectively the description of our experiment. That's the description of all of our samples and the experimental conditions that they fall into. So we're assuming now at this point that we have 4 experimental conditions in this experiment. And this column, which we're going to add into our sample table, describes those conditions for our 12 samples. So it's important to have that in there and to consider that before you create your DESeq dataset. So to add this in to your sample table, you define it as a new column. You can do it with this dollars notation, or you can do it with this double brackets notation. It doesn't really matter which. I'm actually going to use the dollars notation because that's what I'm more used to. So this column doesn't yet exist. So if I just run this, it will give me a null because it doesn't exist. But we're going to make it exist by setting it equal to conditions, and that will add it into our sample table. So now if we again have a look at the sample table, you'll see that that column has now been added at the end. So the first column is our sample IDs, and that's important for reading this in. Those sample IDs in that first column, they map to the column headers in our dataset. They're the same. And then we've got this last column, this conditions column, which describes our experiment. So that's our col data. So sample table is our col data. And the final thing that we need to describe, to load this experiment into DESeq, is this design. And the design here is a mathematical description of the design of our experiment. And the way that you define experimental designs in R is a little bit strange and will be unfamiliar to people, and you won't have seen it before, but you use the tilde character. So essentially this means, what this means is the character, when you think about a design, so I'm going to take myself to a different line of the file, hold on. When you think about a design in R, this tilde sign means given. So what you're doing is you're saying you want to find y given x. So in the case of our experiment here, y is the expression of a gene and x is the design of our experiment. So you want to find y given x. You want to find whether a gene expression, the expression of a gene, is dependent on the design of your experiment. And that's the question that you're asking by setting up your design like this. In DESeq and in Limma as well, the left-hand side of this design equation is implied. So the left-hand side is assumed to be my gene expression matrix, effectively. And the right-hand side then is some condition that you can pull out of your sample table, out of your col data. So I'm going to make my design tilde conditions, because conditions is the column that I have just added to my col data, to my sample table, for exactly this purpose. So that is the design of my experiment. Okay, so I'm interested in finding gene expression relationships that are dependent on the conditions in that column. So I've called it conditions, which is slightly conflating two things, but gene expression that is dependent on the categories of sample in that conditions column. And that's the question I'm trying to ask. So this command here will generate a, I don't know why this cross is here, unmatched opening bracket, weird. This command should, all things being well, create a DESeq dataset for me. So let's try it and see. No, why does that not work? All variables in design formula must be columns in col data. Well, that is a column in my col data because I made it a column in my col data. Weird. What have I done wrong? Error in DESeq dataset, design equals design, ignoring all variables in design formula must be columns in col data. Col data is sample table and contains a column called conditions, but I can't spell conditions. Typo, it's always a typo. There you go. So that should do it. And yes, there you go. So that has created a DESeq dataset. Thanks, Hans. That's created a DESeq dataset, which I should now have in my environment. And indeed, there is. So DESeq dataset, a large DESeq dataset, 60,233 elements, 20.9 megabytes. So this is my now my count data encapsulated in a DESeq dataset. Okay, so I have now not only got my count data into R, I have it in DESeq ready to analyze. What I want to talk about a little bit now is the normalization of this kind of count data. So if we have a look at our count data in DESeq, which we can do it with what's called an accessor method. So there are functions of DESeq which exist to extract data out of this DESeq dataset. So one of those is counts. And counts allows me to extract, as the name suggests, the count data from my DESeq dataset. So it takes an argument, which is the DESeq, the dataset you want to extract the count data from, and this will print out my count matrix. Okay, so this is at least the beginnings of my count matrix that I've imported into DESeq. What you'll notice, the difference between this and the data which we looked at which was the results of TX import yesterday, which was so let's just have a look at some particular observations. Let's look at 1, 2, 6. So the first 6 rows and the first 3 columns of this data, just so it fits on the screen. Okay, so that is the data that we've now imported into DESeq. And then if we look back at the count table yesterday, what did we call it? Count data. The counts in the count data, which is what DESeq has imported. if we look at that data, so the first line here, this is the data in the DESeq dataset, and that's what you can see in the console here. The second line, this is the TX import data that was the results of doing that yesterday. And you can see, hopefully you can see, that the data in DESeq and the TX import data are subtly different. And that's because when you import data into DESeq, it rounds it for you. It rounds it to the nearest integer. There are reasonable reasons for that. DESeq expects to work with integer data, expects to work with count data, which is expects to be integers. And the results of TX import in an ideal world would be integer data, because when you add up all of the counts for a transcript, you should get to a whole number. But the genomics is rarely that straightforward, and the existence of paralogs of genes within an organism means that quite often reads will map to more than one gene. So you will find that you don't end up with integer counts for that kind of reason, that actually the counts for some reads are shared not just between the transcripts within a gene, but actually between genes. So you don't end up with integer counts. So one of the things that DESeq does is just it ensures that you have integers, because that's how it likes to work. This has no material difference really on the results. It's not enough of a change to have any huge effect on the results of what DESeq is going to do. But so what I wanted to talk about in the next 10 minutes or so is whether this is the be all and end all, whether once we've got this data into R like this, that this is what we then go on to analyze. And so the thing that I always do, the question that I always pose when we do courses about RNA-Seq, when we talk about this when I talk about this with students that I'm teaching, or when I talk about it on courses with it, is I show them this example count matrix. I'm showing you this example count matrix, which you've already seen, and I pose the question, which I need to find. Here it is. Are the differences across the columns in this table the result of changes in gene expression? And because I don't have a particularly low latency streaming set up, it's going to take you about you're viewing this on about a 20-second lag from me saying it. I'm going to answer the question myself, although I may have padded for long enough for somebody else to answer it, but I suspect not. So are the differences across the columns in this table the result of gene expression changes? The answer to that is no, they're not. The differences that you're seeing between these columns are reflective of a number of things, and we can't know if they're due to changes in gene expression until we've normalized the data. So until we've managed to apply some sort of normalization procedure. The reason for that is that in an RNA-Seq experiment, reads map to a gene in proportion to three things. So they map to a gene in proportion to the length of that gene, the amount of expression that gene is exhibiting at that particular moment in time, and the depth of our library, that is the number of reads in our library. And of those three things that our counts are dependent on, we're only interested in one, and that's the middle one. We're only interested in the gene expression. So we're not bothered if one gene gets twice as many counts as another gene because it's twice as long. That makes that we're not interested in that most of the time. And actually, for the purposes of finding differential gene expression, the length of the gene doesn't really matter anyway, because you're comparing gene A in condition 1 with gene A in condition 2. And gene A has the same length in condition 1 and in condition 2. So actually, the fact that read counts are variable as a as a function of length doesn't really matter when you're comparing the same gene between conditions. It only matters when you're comparing gene A to gene B, and they might have different lengths and therefore different depths, different counts, even if their expression is equal. But the library depth effect does matter. So if you've sequenced 10 million reads for one of your libraries and 20 million reads for another of your libraries, the second of those libraries will have twice as many counts for every gene, regardless of expression effects, because you've generated more sequencing reads for that library, and so that is the effect that you need to normalize away. That's what the purpose of normalization in RNA-Seq analysis is, and in any kind of analysis, your normalization is an attempt to remove systematic effects that are a result of the way that the experiment was done, rather than biological effects that are a result of the reason that the experiment was done. So the biology that you're interested in. DESeq applies this this methodology for normalization called TMM, or trimmed median of means, for normalization. And on this slide is the basic procedure. So we calculate a per row geometric mean of the counts, and then a per row ratio of the counts to the geometric mean. I'm going to show you an example, so don't worry too much about the maths of this immediately. Then you calculate the median of those ratios for a given column, and that generates you a scaling factor, and then you apply that scaling factor across your whole table. So you divide your raw counts by the scaling factor to give you normalized counts. So just to show you this worked through for that example data set that we were looking at before. So this is that data set, and in particular I want you to pay attention to these two numbers in the top left hand corner. Okay, so 176 and 539. Those are the raw counts for the first gene in this experiment in sample 1 and sample 2. And in this example, there's about a threefold difference between those two samples. So 539 is roughly about 3 times 176. Okay, so let's turn our attention to normalization. So the first thing that we have to do, according to the procedure on the previous slide, is calculate a per row geometric mean. So the geometric mean is the nth root of the product of the observations. So in this case, we've got 6 observations. So you multiply those 6 numbers together, and then you find the 6th root of that product. So 176 times 539 times 569 times 787 times 636 times 681 gives you a very large number, and then you find the 6th root of that number, and the 6th root of that number is 513.81. Okay, so that's the geometric mean of that row, and you do that for every row in your data set. So in our data set, we've got about 60,000 rows, so we will calculate about 60,000 geometric means. So that's the first step. Then the next thing that you do is you calculate the ratio between the count, the observed count in a particular sample, and the geometric mean of the row that it falls in. So that's what these are. Oh, I should note actually, before I move on, the geometric mean of a row that contains any zeros is always 0. Because you're finding a product, and any multiplication involving 0 always gives a 0. So the geometric mean of any row containing a 0, and it doesn't matter what the other counts are, if there's a 0 in the row, then the geometric mean of that row is 0. And for the procedures that follow, those rows are then ignored. Because the next thing that we do is we divide all of our counts by the row's geometric mean, which for that row, which has a geometric mean of 0, results in either not a number, where you're dividing 0 by 0, or infinity, where you're dividing a positive count by 0. So this row is ignored for everything that follows. But for everything else, you generate a ratio between the count your observation for that gene in that sample and the geometric mean of all of the observations for that gene across your whole experiment. And that's what these numbers are here. So these are those ratios of count to geometric mean of the row. And then you go down all of these columns and you find, excluding the rows with NANs and INFs in, you find the median of those observations. So you find the median of those ratios in each of the columns now. So before we worked row-wise, and now we're working column-wise. Okay, so we find the median of the observations in the columns, and that is what we then call our size factor. So the median of the ratios in the columns, and that's the ratio between the count and the geometric mean of the row, is then your size factor for that library. So the size factor for sample 1 is 0.518832. The size factor for sample 2 is 1.611, etc. So these are our size factors. And then to generate our normalized counts, we divide our raw counts. So we go back to the raw numbers and we divide the raw counts by the size factor for the sample that the count belongs in. So we go back to your raw samples and you divide all of the counts for sample 1 by the size factor for sample 1, and that gives you your normalized count table. So this is our normalized count table. So after we apply all of those procedures, we get to these normalized counts. And going back to those two observations in the first two columns, sorry, I've highlighted rather too much, but those two observations in the first two columns, they once you've normalized, no, I'm not going to do that. You can now see, hopefully, that they are essentially the same. So after normalization, before they were about threefold different, so from about 170 to about 540. After normalization, they're both around about 330. And that's because their size factors are about a factor of 3 apart. So the size factor of library 1 is 0.5 and the size factor of library 2 is 1.6. So if you divide one by 0.5 and divide the other by 1.6, you end up bringing them together in the middle. So after normalization, we can see that there is no practical difference between the expression of that first gene in sample 1 and sample 2. The geometric mean, it's a question in the comments, the geometric mean is precisely to remove the effect of those rows which contain zeros. So the rows which contain zeros will otherwise have a very large effect on the normalization. And it's to remove them from consideration, basically. Otherwise, they'll have a significant effect. Geometric mean is more robust to outliers than the standard mean or the median as well. Okay, so that's that. So that's our normalization. This is the procedure that DESeq applies to normalize the count data that we give it. And let me hide that by finding the right key combination. There we go. So in order to apply that to our DESeq dataset, we have to run DESeq dataset equals, and it's estimate size factors, I think. I'm just going to have to make sure I remember this correctly, so I don't want to get this wrong. Bear with me 1 second while I check this. This is a slight lack of prep time today. We've had a high maintenance 7-year-old today. Hence, what feels like a slight lack of preparation from other days, and I would normally have had this open in plenty of time. I'm just going to grab the right document to check that I'm running the right function. So here we go. Normalization. Yes, estimate size factors, I'm right. Okay, so apologies for that. Get rid of that again. Getting better at hiding my dirty laundry from prying out from your eyes, at least. Right, okay, so we estimate the size factors of our DESeq dataset. So estimate size factors takes one argument, which is the DESeq dataset that you want to estimate your size factors for. That will find the size factors for your library. You can find out what the size factors are with another accessor method. So we used the counts accessor method before for getting the counts out of our DESeq dataset. We can get the size factors out by using the size factors accessor method. The one thing I would say is that with TX import, this may not work. So having said previously that, let me find the slide again. Here we are. Having said previously that we didn't care about gene length because we weren't going to be comparing gene A to gene B, so gene length didn't matter. Actually, because we're using TX import and the gene length is in the results of TX import, DESeq does actually use it, does make use of that data. So what we end up with actually is not just size factors, but we end up with a per gene normalization factor as well, which is the which then is encoded in not in size factors but in normalization factors, where it takes the size factor and it moderates it by the gene length. So size factors for this data set is null, and that's because we in fact have normalization factors in this data set, which is a matrix of the same dimensions as our gene expression matrix, and every gene, basically every observation, every gene in every sample has its own normalization factor, and this is the size factor for that library moderated by the length of that gene. So there is a relationship between all of the numbers in the columns here, but they're mitigated by the length of the gene, I suspect in kilobases. This is actually, the normalization that DESeq is actually doing includes a gene length normalization as well as a library depth normalization. Okay, just so you're fully aware of what's going on in this data. If we want to now have a look at the normalized counts as well, we can do that with the counts function that we've already seen. We pass it an additional argument. So counts DESeq dataset normalized with a Z equals true, and that will give me the normalized counts. Again, I'm just going to get the first 6 rows and the first 3 columns so it fits on the screen. So those are my normalized counts, whereas my raw counts look like that. So you can see that in some cases there's not a huge difference between the raw count here 218 and the normalized count here 225, but in other cases there is quite a large difference. It has quite a normalization can have quite a large effect on those